about our recent work on using deep learning for the analysis of cellular electron cryo-tomograms. So this is a painting of a hypothetical subregion of cellular cytoplasm. So inside such a cytoplasm, we can see that there are a lot of macromolecular complexes with complicated structures and spatial organizations. So macromolecular complexes are nanomachines that participate in a wide range of cellular processes. So to fully understand such cellular processes, we need to know systematically the native structures and spatial organizations of all macromolecular complexes inside single cells. So this information has been extremely difficult to obtain, primarily due to the lack of suitable data acquisition techniques. So the recent advance of cellular electron cryo-tomography technique has made it a promising tool for studying the native structure and spatial organizations of large macromolecular complexes inside single cells. So basically, for such imaging technique, the cell sample is first quickly frozen and put into a transmission electron microscope. And then we have the electron beam pass through this cell sample to obtain a projection image of this cell sample. And then we can rotate this cell sample to obtain a series of the projection images. And these projection images can then be used to reconstruct a 3D image of the subcellular structure at submolecular resolution and at close to native state. So this is a slice of such a 3D tomogram that contains a small bacterial cells, a bacterial cell. And in principle, such tomogram captures the structure and the spatial organization of all large macromolecular complexes inside this field of view. However, the analysis of these complexes is extremely difficult because of several reasons. So first, there are a high level of imaging limits because of the missing data and the low signal-to-noise ratio. Secondly, there is a high structural content complexity because the macromolecules structures are highly heterogeneous, and also they are often inside a highly crowded molecular environment. And in addition, there is a big data issue. So nowadays, with the advance of the automation of this data acquisition, it's not difficult for electron microscope to capture hundreds of CECT images within several days. And each image can have 16 billion voxels in terms of their size. And also, these images could contain more than 1 million macromolecules. And these macromolecules are highly heterogenic, and they could contain at least hundreds of structure classes. So, therefore, to efficient and accurate separate or filter these macromolecules into subsets of structurally homogenic macromolecules, it's very critical for the systematic recovery of the structures of these macromolecules. So in such tomograms, the macromolecule is represented as a subtomogram. A subtomogram is a small cubic region. A subtomogram is a small cubic region cut out from this large tomogram that is likely to contain a single macromolecule. So Over the past years, we have developed a number of techniques to analyze such subtomograms, including structural pattern pursuit. Including structural pattern pursuit, which is a constraint optimization approach for the structural pattern mining, and also a different type of subtomogram alignment and segmentation and coarse separation techniques. In addition, we also developed the tomogram simulation and also open source software platform, including computer cloud support. Specifically, in order to separate these subtomograms, we and others have developed reference-free subtomogram classification, averaging, and pattern mining approaches. However, such approach involves a subtomogram alignment step or integration over exhaustively enumeration of the rotation operations or rate transformation of operations, which is computationally intensive. Therefore, such type of approach is only capable of processing tens of thousands of subtomograms using a computer cluster. So in order to increase the scalability, we also developed a rotational invariant feature and post-normalization approaches for the coarse separation of subtomograms. However, such approaches has a limited accuracy because they do not take into account the missing data effect, and also the post-normalization approach has a high dimensionality problem. So in this project, to complement existing approaches, we develop a 3D convolutional neural network approach for a supervised classification. So given an input subtomogram, we use convolutional neural network to apply stacked convolution operations to extract the image features from this subtomogram, and use these image features to determine the structure class of this input subtomogram. And this convolution kernels are learned so that they capture the complex mapping between this input subtomogram and the output class label. These are two network architectures that we have developed which are adapted from the popular architectures used in the computer vision community. So, of course, they normally use 2D filters, but in our case, we extend these 2D filters into 3D filters. And the first architecture is characterized by an inception model, which is a parallelization of several convolutional filters with different kernel size, which captures the image features at different scale. The second one is characterized with a deeper architecture and small convolutional kernels. So, and once these classifiers are trained, we remove the last softmax activation layer to obtain a more continuous representation of the class assignment, which can be treated as a structural feature extraction, which projects the input subtomogram into a low-dimensional structural feature space that is spanned by the training classes. So, such structural features are expected to be invariant to the radius transformations and also the missing data effects. And our test shows that these features are more robust to noise, and I will show you in the results section. And they are also highly scalable. Specifically, once these classifiers are trained, we can extract a million subtomograms using a single workstation equipped with 2 GPUs, and within 1 hour. Okay, so we have tested our methods on both the simulated and experimental data. So we first constructed simulated data sets, and each data set has a specific signal-to-noise ratio and tilt angle range setting. And for each data set, it contains 23 classes. And for each class consists of 1,000 simulated tomograms with randomly oriented macromolecules. And this data set is split into a training set and a testing set. So this is the prediction accuracy. We can see that for our new approach has significantly achieved a significant increase of the classification accuracy compared to our previous method, which is a rotational invariant feature-based method. So in addition, because in most of cases we do not know the native structures of macromolecular complex, so it is very important for an approach to be able to detect new structures that does not exist in the training data. So to test this, we first removed the proteasome subtomograms from the training data, and then project the test data into this feature space. And this test data includes both proteasome and other complexes. So we found that this proteasome subtomograms are still enriched in certain subregions in this feature space. So in such case, if we over-partition these test samples using clustering, so we are able to obtain clusters that where this proteasome are highly enriched. And by applying our previously developed classification and unsupervised classification and averaging techniques, we may expect to recover the structure of this proteasome, even if it does not exist in our training data. So to systematically test this idea, we have performed a leave one out test, and we are able to recover 16 of this macromolecular complex structures. So in summary, cellular electron cryo-tomography is probably among the highest resolution 3D imaging techniques for imaging the intact and close to native state subcellular structures. And nowadays, it's possible for CECT to capture millions of macromolecules, and these macromolecules are highly heterogenic. So therefore, efficient and accurate structure separation is the key toward systematic structure recovery of these large macromolecular complexes. So we in this project, we formulate this problem as a large-scale supervised 3D image feature extraction problem, and we developed a deep learning approach which outperforms our previous approaches in terms of speed and accuracy. So, and as most of the structures of macromolecular complex are known, so we demonstrate that in principle, we are able to recover unknown structures that do not exist in training data. Therefore, our supervised approach in combination with unsupervised approach is able to recover unknown structures. So, and we also have some new progress. So we have recently developed 3 new network models that has even higher classification accuracy compared to these previous 2 models. And also, to complement this supervised approach, we also developed an unsupervised convolutional autoencoder approach for the characterizing subtomograms and image features. We furthermore extended this autoencoder approach for weakly supervised coarse segmentation of the cellular structure and components. And So I'd like to thank Dr. Murphy for suggestions and also support from NIH and Winters Foundation. So, and I will be also at this afternoon's poster session if you'd like to know more details about our projects. Thank you.
Thank you very much. Questions? Can you approach the mic?
So I have a question concerning the deep learning model. So if you don't know how many structures in your data set, how do you determine the number of nodes in the last layer, softmax layer?
So in the training data, we need to know the number of classes.
Okay, so so in the test data, you have some unknown structures. Right. So you would do the use the deep learning to extract the feature, and then do unsupervised learning to do the clustering, right?
Yeah, right. Actually, yes, of course, there is a problem of determining the optimal number of the clusters, yes. Which is, I think, to us, is still an open problem that we need to look at.
Yeah, thank you.
No problem.
Can you go back to the slide where you showed which ones you did recover and which, yeah. Looking at this slide, can you tell us as to where the field is going, which ones are easier to find and more difficult? Is it size, resolution, shape, complexity?
I think that's a very good question, and however, we haven't looked at this aspect yet. Yeah. And currently, we only focused on improving the classification accuracy. However, your question is certainly a very important direction that for us to have a close look. Yeah, right. Thank you.
Thank you, Min.
Okay.
And thank you.